smooth muscle, it's a 12-to-1 ratio, but it's much less weight than we see. Calcium is involved in the regulation of contraction, just like in skeletal and cardiac, but it does it in a different way. And we'll look at that way. But we do sequester some calcium, but they're in these little membrane vesicles, bound vesicles called calveoli, and that is only found in smooth muscle. There is no transverse tubules. The cells are so much smaller. You don't need to have the perpendicular or transverse tubules as part of your conducting system. So there's no diads, there's no triads, there's nothing like that. When we look at smooth muscle is in these three major places. The first is the viscera. The viscera simply means the internal body organs. And in the walls of most of our organs and ducts and tracts, we have smooth muscle. And that smooth muscle functions to move things through from proximal to distal. It functions to, like in the stomach, grind things up. In the urinary bladder, it helps push the urine out during urination. During labor and delivery, it helps push the baby out of the uterus. All of this is smooth muscle. And so we have lots and lots of smooth muscle in the viscera. We'll see a little bit today in lab, but even more when we get to the digestive system and others, that very often when the smooth muscle is lining a tract, it's in at least and most commonly 2 layers, an inner layer and an outer layer. So we're going to see that. So we're going to be looking at smooth muscle very often in 2 different orientations, running longitudinally and running cross-section. And so we'll look at that some today as well. Now, this is another interesting thing, is a lot of the smooth muscle in the viscera has what we call a functional syncytium. What that means is, like the heart, you can have a pacemaker and the ions can move through from cell to cell by gap junctions, and it can contract as a unit. However, in smooth muscle, the contractions are much slower and more sustained, longer duration than in cardiac. Cardiac, you have to contract it, move the blood out, then relax it, let the blood in, and repeat. In smooth muscle, a lot of times you have a sustained contraction that will go on for some time. This is called the single unit or unity smooth muscle, and so is the vascular. The multi-unit is in the iris. So this is kind of what we have here. Now the vascular smooth muscle, it differentiates in situ, just means in place, from mesenchyme. Again, these are both, the first 2 are mesoderm derived, and they come from mesenchyme and then on into the maturity. And this vascular smooth muscle is in the walls of most of our blood vessels. Some of them are too small, like capillaries, but most of our blood vessels have smooth muscle in their wall. When we look at cardiovascular, we look at the blood vessels, and we'll see that there is different layers. There's a tunica intima, there's a tunica media, and a tunica adventitia. Well, the tunica media, the middle layer, is where you have your smooth muscle. And of course, the purpose is to vasoconstrict with contraction or vasodilate with relaxation, so you can control blood flow and blood pressure. And so that's all smooth muscle. Here, the waves of contraction are more localized to that particular area, and they're not sustained like they are in visceral smooth muscle. So there's a difference there. finally, the smooth muscle of the iris is derived from ectoderm, and each smooth muscle contracts individually. That's why we call it a multi-unit rather than contracting as a unit. So instead of a single unit contraction, it's a multi-unit, different cells contracting individually. Here is some of the so the diameter, let's say around 5 microns, and maybe you're about 200 microns in length. And notice the skeletal muscles may be about, well, let's just say about 50 microns wide and tens of centimeters long. So you can see there's a huge difference between the length of the smooth muscles and the skeletal muscles. Smooth muscles have a single centrally located nucleus, whereas of course skeletal muscle is multinucleated, and the nuclei are hypolemally or under the membrane or at the periphery, all sort of saying the same thing. Smooth muscle does have myosin and actin and tropomyosin, but no troponin. So there's no regulatory troponin protein there. When you have your actin, it can either be anchored to proteins in the plasma membrane or in the cytoplasm to structures called dense bodies. So the dense bodies are also unique to smooth muscle. There's no sarcomeres, as we've said. It does contract by a similar sliding filament mechanism, but it's not arranged near so neatly to form a banded pattern. Again, you can have these gap junctions in most smooth muscle, which allows ions to move from cell to cell and contract multiple smooth muscle fibers as a unit. You do form little fascicles, that's bundles of nerve fibers in smooth muscle, but they're not near as big. They'll have paramecium around the bundles and epimecium around the entire muscle. This shows a contraction and a relaxation. Now, as we look at the actual images, here is the light microscopy image, and here is the electron microscope image. These are smooth muscle, these are the nuclei. You notice that the smooth muscle are tightly packed in there together, so they're pretty well packed in there. This layer is running longitudinally as we're looking left to right. This layer is smooth muscle that's coming at us. It's perpendicular, it's cross-section. So here's a nucleus and a fiber around it. Here's a nucleus and a fiber, and all you can see the nuclei are centrally located there. We'll have a little bit of connective tissue around that supports it. Notice this is a pretty high magnification, by the way. This is 600. So a little higher than our typical, what we see under 400 is our typical higher magnification. Now, this is the two layers that we often see in the walls lining tracts like the digestive tract. Here's a longitudinal layer, and here's a cross-sectional and transverse layer. What we're going to see, and we're going to be starting the nervous system, this is a ganglion between the two muscle layers. This is called a myenteric plexus, or it can be called an Arbox plexus. It's a parasympathetic ganglion. Unfortunately, it has more than one name, but these are neuronal cells, ganglion cells, and neuronal fibers in between these two layers. muscle. So we'll be seeing that quite a bit. So here's our muscle cells here, and then here in cross section, a little bit of connective tissue out here. Here's another showing some smooth muscle cells. Again, this is cross section here, and this is longitudinal here. Now this is a pretty image, an electron micrograph, and here's the longitudinal from about here down, and here's cross section. Now look at, here's a nucleus, and then here's the cell membrane and the muscle fiber. There's another nucleus, another one. See how they're packed in there, all of these. Like here's a muscle cell, here's another one, here's another one, here's another one. And they're running. See how it kind of tapers? You know, it looks better than my drawing here. It tapers around there. And then over here we see them in cross section. And so here's the nucleus and the nucleolus, and the cross section. You can see the cell membrane in this electron micrograph. This is simply a capillary, little tiny blood vessel running through. The muscle cells are very well vascularized, but cardiac and skeletal are more highly vascularized than smooth muscle because they require more energy. Cardiac is especially vascularized.
How can we differentiate in like the transverse cut between smooth and cardiac? Because they both have the nucleus pretty central.
You'd have to see something surrounding. You'd have to be able to see enough to see it in longitudinal as well.
Okay.
Because you couldn't see any striations in the cardiac.
Or the disc.
And you couldn't see any intercalated disc. And so you wouldn't really be able to identify it. If this is all the information you had, all of this was obscured, and then you say, okay, what is this? That wouldn't be enough.
Okay.
We'd have to know more. Yeah. This shows it. With the longitudinal. And you can see these calveoli, those vesicles that contain calcium. Now, for contraction of smooth muscle, we use enzymes. We phosphorylate and dephosphorylate the myosin, and that phosphorylation and dephosphorylation is controlled by two different enzymes, and it's regulated by the calcium concentration. The easiest way to do it is to look at this diagram. If you increase cytosolic calcium, it will bind to calmodulin and activate it, and that will activate inactive myosin, the light chain kinase, that's an enzyme, as the ASE alerts us. So it takes inactive myosin light chain kinase and activates it. That puts then causes the phosphate, the cross bridge of the myosin, once it gets phosphorylated, to bind to the actin. To unhook it, we dephosphorylate it, and we use myosin light chain phosphatase instead of a kinase, and that takes the phosphate off, and that takes the heavy myromyosin or the myosin head off, and so it disengages. Increased calcium in the sarcoplasm activates this enzyme, decreased calcium activates this enzyme. And so you trigger it with calcium, but through an enzyme, not through troponin and all that. So there are some of the differences there that we see. Another difference is calcium here doesn't just come from the sarcoplasmic reticulum. It can, but it can also come from extracellular calcium through the membrane, the sarcolemma. So you can get two sources for calcium. This is a comparison, and again, we're not going to give you much on this physiology, but if you want to remind yourselves of how calcium is used to control contraction in smooth muscle, that's shown on the left, and how it's done in skeletal muscle, that's shown on the right. So there's some comparison there between Now, we're moving away from smooth muscle per se, and we're looking at just a few examples of muscle injury, and this can include all three types of muscle. We can have an injury of skeletal muscle, and a lot of times these injuries are the connective tissue associated with the skeletal muscle. The connective tissue can regenerate, and you can have some immature stem cells that can fuse to form some new skeletal muscle fibers, but this is quite limited. Mature muscle fibers cannot undergo mitosis. You can only get new ones from stem cells that then differentiate, and that's limited. How you increase muscle size and strength is to add protein inside of existing skeletal muscle cells, fibers. Cardiac muscle also has little regenerative ability, and therefore if you have a myocardial infarction, you cannot regenerate those dead heart muscle cells. You can make surrounding ones stronger, but you can't regenerate it. However, smooth muscle can undergo mitosis. It can multiply, and in fact, it does multiply in the uterus. The smooth muscle lining of the uterus is the myometrium, and that gets bigger when we have in pregnancy. We can have myoepithelial cells in different areas, and they have some contractile ability. For instance, they can help push sweat out of sweat glands and other oil, ducts, sebaceous glands, and that kind of thing. They can push milk out of the glands of the breast. They can push tears out of the lacrimal glands. And so we're using a number of different individual cells that have contractile ability. These, again, arise from ectoderm instead of mesoderm, so there's a little difference there. If we have Duchenne muscular dystrophy, what happens here is there's a genetic defect that causes us not to produce a protein called dystrophin. That's why it's called dystrophy, and that helps connect the muscle proteins to the sarcolemma, and so it means that the muscle is weakened and destabilized if you don't have enough dystrophin. Most of the cases of dystrophy, including Duchenne muscular dystrophy, the most common are on the gene is on the X chromosome. So they affect males at a much higher rate than females. It's an X-linked, and this can cause death before the age of 20. Some individuals live up to 30 or so, but oftentimes death occurs before that. It can affect both skeletal and cardiac muscle, and the skeletal muscle issues usually result in death because you can't breathe, you can't expand your lungs, the skeletal muscles. The cardiac muscle, of course, would result in inability to pump enough blood. So that's why it generally causes death. ALS, or its somewhat more common name, Lou Gehrig's disease, because of a baseball player years ago now who died of it. It was also named for him, Lou Gehrig, but it's amyotrophic lateral sclerosis. This is a problem of the motor neurons in the spinal cord not degenerating. So then they don't activate the muscles, and the muscles atrophy. And again, death is usually due to respiratory muscle failure. Myasthenia gravis is an autoimmune disease where the antibodies that you have the receptors that acetylcholine should bind to, where you make some antibodies that block to it, but they don't activate the muscle contraction, but they block the ability of acetylcholine to bind. And so this can be a problem. You're generally going to try to treat this by maybe removing plasma temporarily and taking out these antibodies and then reintroducing it by using immunosuppressants to try to decrease the activity, things of that nature. Myocardial infarction, again, usually there's ischemia to an area, a lack of blood flow to part of the heart that causes the cardiac muscle cells to die, which is an infarct, an infarction, and that can cause death, of course, or something less, but weakening. Rigor mortis, you know, because ATP is also needed to disengage the myosin cross bridge from the actin. If you, after death, you don't have enough ATP to do that, and so for a while, the muscles can stay contracted, the muscles that were contracted. Eventually, those proteins will break down in a few hours, but at first there will be this contraction. And then botulism, that inhibits acetylcholine release at the neuromuscular junction and can result in death if it's too severe. Oh my, well that was. So we'll introduce the nervous system here in the next few minutes, and then we'll continue on some at 2 o'clock over in Clark Hall 201, and then of course we'll do our muscle lab. We actually are going to have a partial lecture today on the nervous system, one on Friday on the audio lecture, and one next Monday. So we have several days to go through this. First of all, you're well aware that to make it convenient, we divide the nervous system into the central nervous system and the peripheral nervous system. The central nervous system, you remember, is the brain and the spinal cord. The peripheral nervous system involves all the peripheral nerves and the ganglia. All the peripheral nerves are derived from two sources: the cranial nerves, which are peripheral nerves, but they're attached to the brain, so they're called cranial nerves, and there's 12 pairs of cranial nerves. You remember these: olfactory, optic, oculomotor, etc., right? Those are the cranial nerves. The spinal nerves are called spinal nerves because they attach to the spinal cord, and there's 31 pairs. So there's some that attach in the neck area, they're called the cervical, and then the thoracic, the lumbar, the sacral, the coccygeal, 31 pairs. All the spinal nerves are mixed, they're both sensory and motor. And then all the other nerves are derivatives of the spinal nerves. They branch and then you call them by a different name. We have two major types of cells in the nervous system. The neurons are the ones that conduct electrical information. And then the glial cells, or the supporting cells, are called neuroglial or glial cells. And they're supporting, in a supporting role. You remember in the central nervous system, we have this big cerebrum, which is about 80% of the brain. We have the diencephalon with the thalamus and hypothalamus, the midbrain, the pons. Maybe you can see it better. Turn this on. Cerebrum, diencephalon, midbrain, pons, medulla oblongata, cerebellum, spinal cord. These are the different regions. Here's your cervical nerves, 31 pair. This is your spinal cord. The inner area is gray matter. That's neuronal cell bodies and dendrites and the like, primarily. That's the H, or the butterfly shape. And the outer region is the white matter. That's mostly axons, mostly myelinated axons. We have the front and back of the spinal cord, except instead of calling it front and back, we call it anterior and posterior, or ventral and dorsal. So ventral here and dorsal to the back. Here's the spinal nerves. Every spinal nerve, all 31 pair, when it's about to attach to the spinal cord, it branches into two roots, a dorsal root and a ventral root. The dorsal root carries sensory information into the spinal cord. The ventral root carries motor information away from the spinal cord. There's a little swelling, a little ganglion on the dorsal root. That's called the dorsal root ganglion. That's where the cell bodies for most sensory neurons are located. So they're located right there in that little dorsal root ganglion. Of course, the vertebrae protect the spinal cord there. This shows the neuron, or the dendrites, carrying information into the cell body. And here's the cell body, also called the soma, or the perikaryon. Here's the nucleus and the nucleolus. In the nervous system, the neurons, the endoplasmic reticulum, the rough endoplasmic reticulum, and the ribosomes stain in a unique way. And so we call this rough ER and ribosomes the nesal substance, or the nesal bodies. And so we'll see that in staining. Then we have the single axon, which can branch, and it can be myelinated or unmyelinated. So here's the central and peripheral nervous system. Sensory is carrying information from the sensory receptors along sensory axons, neurons, to sensory nerves, towards the central nervous system. Motor is carrying it from the central nervous system to the effectors. The effectors are things like muscles, glands, and organs. In the somatic nervous system, it's voluntarily controlled, and the effectors are the skeletal muscles. In the autonomic nervous system, its effectors are involuntarily controlled, like cardiac muscle, smooth muscle, and glands, and it's going to be involuntarily controlled. So a little bit of differences there. Again, the neurons conduct the impulses, electrical activity. The glial cells support, nurture, and protect the neurons. We have probably 5 to 10 times more glial cells than neurons. There's more support cells. Just like in muscle, the nerves have connective tissue around them. The outermost connective tissue layer around the nerve is called the epineurium. This is analogous to around the muscle, it's called the epimysium. Then we have bundles of nerve fibers. Those are called fascicles of nerves, axons for instance, and around there you have connective tissue called perineurium, which is analogous to paramysium. And around each individual axon you have connective tissue, which is called endoneurium. And again, that's analogous to endomysium, or in the muscle. Neurons can have different shapes, or can look differently. Some are called unipolar or pseudo-unipolar neurons. These make up most of our sensory neurons, the sensory neurons that are carrying information towards the central nervous system. And their cell body is located primarily in this dorsal root ganglion. So the cell body here is located in the dorsal root ganglion primarily. We have a couple of examples in the body of bipolar neurons. Some are in the retina of the eye, some are in the olfactory area, our interneurons, which are all housed within the central nervous system, or our motor neurons. They have this multi-unit shape with a number of dendrites, cell body, and a single axon which can branch, and so they look more like this. This shows that classification again. Notice pseudo-unipolar and unipolar, that term is often used interchangeably. This is a typical neuron with the dendrites gathering information, the cell body or soma or perikaryon, the nucleus with the nucleolus, the axon which can be myelinated as shown here or unmyelinated, and then the axon terminal or bouton. This shows the nuclear envelope of the nucleus and the nucleolus, and then processes coming off here. In the periphery, the axons are myelinated by Schwann cells. That's a type of neuroglial cell that myelinates in the periphery. In the central nervous system, the oligodendrocytes are the glial cells that myelinate the axons. So here are axons in cross-section. It's interesting to note that we have some mitochondria shown here. And here's an axon with mitochondria. And here's a Schwann cell with its nucleus, and it's myelinating these axons. Sensory neurons carry information towards the central nervous system. Interneurons are housed entirely within the central nervous system, and motor neurons conduct information from the central nervous system to the effectors. Here are some alternate names for the nucleus, or the cell body, excuse me, the soma and the perikaryon. And the nucleus is usually quite prominent, pale staining, but often has a dark staining nucleolus. Sometimes the nucleolus is described as owl eye. The nasal bodies are the ribosomes and the rough ER. Because of their unique features, they stain in a certain way. They stain dark and under light microscopy. You have other common organelles like the Golgi complex. You can have melanin-containing granules, lipofuscin. This lipofuscin, again, usually stains brown, and it gets more with age. It's kind of metabolic waste products, if you will. We have 3 different types of cytoskeletal components in the neurons. We have neurofilaments, which are intermediate in size. We have microtubules, which are the largest in size. This is nanometers, as you can see. And we have microfilaments, which are actin, and that's the smallest in diameter. Dendrites are going to gather information, and they can have little spines on their surface, which increase surface area. Axons, in most cases, conduct information, impulses away from the cell body. The axon, even if it's very thin, can be very long. For instance, we have axons running from our feet up to our spinal cord, or the other direction as well, from our spinal cord to our feet. So we can have axons that are 3 feet long, for instance. Giraffes can have axons which are like 3 meters long. These axons are very small in diameter, though. You can't see the axon with the unaided eye. You need a microscope. But bundles of axons make up the nerve. So if you have 100 or 1,000 axons all bundled together, that makes up the nerve. So when you see a nerve, it's ulnar nerve or radial nerve or femoral nerve, sciatic nerve, those are bundles of axons. The cell body for all of those axons If it's in the sensory system, it's usually gonna be in the dorsal ganglion. If it's in the motor, it's gonna be in the central nervous system. In the nerve, the only nuclei you're going to see is the supporting cells, the Schwann cells that are gonna myelinate, and you can see fibroblasts that are going to be part of the connective tissue. You don't see the nucleus of the cells, of the neuronal cells. So that's a little bit a unique aspect of it. Just like in muscle, you can have some alternate names. The cytoplasm and the axon can be called the axial plasm. The plasma membrane can be called the axial lemma. So, little differences there. Here's a neuronal structure. This is the nucleus, fairly pale, large. This is the nucleolus, and this is the nasal substance here. This is some processes coming out. There's the nucleolus. This is a model with the nucleus and nucleolus and the nasal substance, and the origin of the axon, the beginning is the axon hillock. These are dendritic spines in the model. This is myelin and the axon here. You see all this dark purplish stuff? That's the nasal substance. And here, and here. This can be called nasal bodies or nasal substance. Here's the nucleus, fairly pale, and the dark, intense nucleolus that we see there. Again, the same image. This is the nasal substance, rough ER and ribosomes that are grouped together. Here we see the nucleus and the nucleolus and the nasal substance that we see staining here as well. Here is some processes coming out. Here's another process. What are all these smaller cells, the nuclei? These are the glial cells. So we'll get acquainted with those eventually. But these large cells here are the neuronal cells, and these are glial cells. These are nerve fibers running through that we see here. We use a number of different stains to look at these. Golgi stains are helpful because the Golgi, for reasons we still don't know, it was developed by an Italian named Golgi. It stains the entire cell, like the entire neuron, but it only stains a subset of the neurons. So if you have 1,000 neurons in the area, it may only stain 100 of them. Well, that is actually helpful because if it stained them all, it would just look like a black mass. You couldn't tell anything. But if it only stains some of them, you're actually able to see them. You can also use fluorescent dyes like this, and you can look at the processes, labeled enzymes, nasal stains, and this shows you the different type of staining that we have. A Weigart stain is a new one that we want to introduce to you. Again, it's a German origin, so even though it's W, it's pronounced like Weigart. And it stains the myelin. If you just use H&E, the myelin won't stain very well. If you use a Weigart stain, it'll stain it black. It'll stain the myelin black, so it depends if you're using that or not. Here's the dendrites with their dendritic spines associated with them. Okay, so for lab today, I don't think we'll actually need, I think we went far enough that we don't really actually need to, there's 2 or 3 I didn't go over that you can go over on your own, but for lab today, I think we can go right into the lab room at 2 o'clock. If I'm a little late from